All right, so continuing on with our module on viruses, we'll next talk about the human immunodeficiency virus, which you've likely heard called HIV. The history of this, the first case was found in the Belgian Congo, which is now the Democratic Republic of Congo, and it was a man who died with the constellation of symptoms in 1959. They didn't know what caused it, but that constellation of symptoms soon became known as HIV and AIDS. The first published report in the United States of a case was in the CDC's Morbidity and Mortality Weekly Report, published in June of 1981. And the MMWR Weekly is what the CDC publishes each week, and it tracks all the different kinds of reportable diseases. So I used to subscribe to this, but now it's all online. So you too can check this if you want to just bookmark that site down there in green, MMWR Weekly, and you can track how many cases there are in the United States each week of influenza, measles, mumps, rabies, HIV, all of those kinds of things. So all the reportable diseases are included in that CDC report. So the origin of HIV, the HIV virus came from what was called the simian immunodeficiency virus. And this was, I wouldn't say it was common, but we don't hang out with chimps a lot, so it's hard to say really how common it is in the wild. But the likely cause of HIV was people in Africa often hunt chimpanzees, and when they were slaughtering the chimpanzee, it's likely that there was a cut in someone's hand or fingers or something, and the chimp who had this virus was being butchered, and the virus was transmitted into the person via the chimpanzee's blood. And it's interesting, I have 35 million humans that, a simple chimpanzee virus has killed 35 million humans. So in the early 1980s was when the disease first appeared in the United States, and there was so little known about this disease. There was no agreed upon name. Different people called it different things. The cause was unknown. There weren't any tests for it. And because it was unknown, what usually happened is patients got super sick. They were ill for months, or in some cases years, but typically months or at least weeks. And they would come in and they would just be so sick. They'd have several diseases, tuberculosis and cancer and whatever else. And they would die from those particular diseases. It wasn't actually the HIV virus that killed them. It was that group of other diseases. So HIV was first seen in men who had sex with men. So originally it was called gay-related immune deficiency, or GRID, because at first it was thought it was just seen in the gay population. And the public and policymakers, when this first started showing up, at that time in the 1980s, there wasn't really a gay rights movement. It was, people were still in the closet, and, it just wasn't given very much attention as far as funding and, and anything that would help to increase the knowledge about the disease. And in fact, in 1983, a doctor in New York was threatened with eviction for treating HIV patients. So in his building, they were like, get these people out of here. So that's a very unfortunate fact of our history. Later on, it was discovered that people who injected drugs also came down with this condition. And obviously, it was very common to share needles among drug users. Needles were not always easy to get. But now things have changed, because in many states there are legalized syringe exchange programs. So if you have a used syringe, you can turn it in and you can get another one or several more. And this type of program has reduced HIV from 50% of drug users in 1990 to 17% in 2002. There's still a ways to go with this. A lot of states are not, in favor of this. They think it promotes drug use, but in fact it doesn't. But it does help prevent AIDS and HIV. So then also later on, it was found in sex workers and still in hemophiliacs. Hemophiliacs use a lot of blood products. They get blood transfusions, or more commonly, they get pooled blood that has certain factors in it that they need. So they'll get small portions of blood from, say, 50 people. And it becomes more common that you would get HIV the more people or blood parts components that you are exposed to. And then finally, it was discovered that mothers who had HIV could transmit it to their fetus in utero, and so babies would be born with HIV. So because of this population that first showed up with HIV, excluding probably hemophiliacs, but it was primarily sex workers, gay men, and drug users. So there was a lot of discrimination and prejudice against that, and so there again wasn't a lot of public policy and funding available for HIV research. So in September of 1982, so the disease has been around for about a year, the CDC labeled the condition, and this is different from the virus, the condition as acquired immune deficiency syndrome, or AIDS. In 1984, so at this point, they still didn't know what caused it, but when you started showing up with several different diseases and you had a very weak immune system, they would label that AIDS. Then in 1984, there were several researchers around the world who were working on this, and three in different countries identified a virus that they named different things, but eventually settled on human immunodeficiency virus. And then in 1985, so 4 years after the first case, there was a test developed for HIV. So you could identify people who had the virus either with symptoms or even previously to having symptoms, which was very important to help in treating or preventing transmission. So what is HIV? HIV in 1983 and 1984 was, as I mentioned, discovered and identified. It comes from the virus family retrovirus, the genus lentivirus, and that particular genus causes chronic and deadly diseases with very long incubation periods, and you'll see that this is important in HIV as well. So the species of these are bovine, which is cow, immunodeficiency virus, equine, which is horse, immunodeficiency, feline, immunodeficiency, cats, human, and simian, which is the chimpanzee version. There's a couple of others that I didn't list just because they're uncommon, and even the animals are uncommon. So what does HIV look like? So if you will look at the picture, you can see that it has spikes, and those are glycoproteins, and they are on the viral envelope. We talked about an envelope. And then just inside the viral envelope is what's called a capsid. And the capsid, it's not like a cell membrane, but it's something that does enclose the DNA and some enzymes together, so that they can remain in the cell and they're not lost, if you will. So those are the most important parts. It's an envelope, spherical, spiked virus, and the nucleic acid found inside is not DNA, it's RNA. So that's an important thing to note too. So within this capsid are the instructions for RNA to use an enzyme, which is found in the cell, called reverse transcriptase. And this will eventually make make DNA. So let's look, well, I'll talk about that again. If we don't get to it, I think I have another slide on that, but I will talk about it again. But let me just mention first, the HIV virus gets into your cell, and then the enzyme reverse transcriptase actually makes a copy of, makes DNA from the RNA template. Okay, so we'll talk about that again in a moment, I'm sure. So there are several virus subtypes. The most common HIV is HIV-1. It's 95% of infections. It's the more serious version. HIV-2 is in 5% of infections, and it's less virulent. It's not quite as bad. And then there's a ton of subgroups of all of this. So there's 4 main HIV-1 groups, M, N, O, and P. M is the most common, and then there are subtypes, A, B, C, D, E, F, G, H. You don't need to know all of those. The main thing to know is that there is HIV-1 and HIV-2, and HIV-1 is the more virulent. This is what the little bugger looks like. Electron micrograph on the left, you can see the spikes on the kind of fainter one, and again on some of the other ones. You can even see the kind of maybe the polygon or something inside, which is the capsid. And then on the right slide, the colorized one, you can see the protein spikes, which are glycoprotein. Oops. I'll talk about that later. So the infection process, the HIV virus is, it gets into your bloodstream, and then what it does is it finds a cell that it wants to infect, which is a macrophage or a lymphocyte cell. And as I mentioned just a moment ago, within the envelope and within the capsid, there is an enzyme which is called reverse transcriptase. And once this virus is in your cell, then this reverse transcriptase makes DNA from its RNA template. So the reverse transcriptase takes proteins and it builds them into a DNA molecule in a certain arrangement as dictated by the RNA template. Okay. So then once it makes its viral DNA, then the viral DNA goes into our cell nucleus, and that small snippet of DNA gets inserted into our DNA. So now the virus has plugged in some of its own components into our cell. And what it does at this stage is it may reproduce some, but it's mostly just in a latent stage. It's not actively reproducing, and it's not actively doing lots of damage. So here's another image of that. You can see the HIV envelope, the viral envelope, and you can see at the bottom the glycoproteins. And then you can see the capsid, which is in purple, and also the HIV RNA. That's that spherical or curly Q strand of nucleic acid. And then it's pointing out the HIV enzymes, which are the reverse transcriptase. Now, we need to go back to the immune system for just a moment. Now, remember, this is human immunodeficiency virus, and immuno means immune, and deficiency means lack of. So if we look at our immune system that we talked about earlier, we have the immune system, and then on the left, we have the innate or nonspecific immune system, and we have the adaptive or the specific. And you'll recall that the specific has two components, as the innate does. It has cellular and it has humoral components, right? So in our immune system, we have two different types of cells, two different types of lymphocytes. So some lymphocytes are identified as T cells, and those are macrophages and other cells, which I'll talk about in a moment. And then there are B cells, and the B cells are what make antibodies. Okay, so they're involved more in the humoral components or the liquid components, making of the antibodies, which are found in our plasma. But the T cells are important in HIV. So here, just again, lymphocytes, there are T and B cells or T and B lymphocytes, and they're involved with acquired or specific immunity. The B cells, as I mentioned, they make antibodies. So what they do is when they see an antigen, they transform into plasmacytes, and then they produce antibodies. You'll probably recall that from our second module. So cell immunity depends solely on the T cells. So certain T cells stimulate other cells, such as macrophages, and then the B lymphocytes too, and other T lymphocytes to fight infection. So those stimulating cells, the stimulating T cells, are called CD4 lymphocytes. Okay, and they manufacture a CD4 protein, and they have a receptor on their cell membrane as well. So CD4 cells help to coordinate the immune response. But what HIV does is it attacks the CD4 cells and destroys them. So when you have the captain of the ship destroyed, it's hard to have an immune response. So therefore, human immunodeficiency. So what it does, HIV enters a CD4 cell, and then it makes hundreds, if not thousands, of copies of itself. Those copies spread out. And once those copies have been made, HIV will kill that original CD4 cell, and those new HIV copies will find new CD4 cells, and they'll restart the signal all over again, or the cycle all over again. And eventually, we have very, very few CD4 cells. So it goes from something like, oh gosh, I don't know, I can't remember exactly, like 5,000 down to 200. And when it gets that low, that's when you start getting AIDS, which is the symptoms. So HIV weakens the immunity against opportunistic. That means those who have an opportunity to infect. They don't all normally infect, but they have this opportunity to now infect because we don't have a functioning immune system. So what are some of those diseases? Well, there's lots of them, but some of the more serious ones are tuberculosis, certain fungal infections, bacterial infections, and even cancer. You might have heard of Kaposi sarcoma. That's a very common one in HIV, but it's very uncommon in the general population. So when you first get infected with HIV, and this is like almost before the dormancy stage, so to speak, you'll have symptoms about 2 to 4 weeks after your infection. You'll get fever, chills, rash, night sweats, muscle aches, just like any other infection. You might also have a sore throat, be tired, have swollen lymph nodes, and maybe even some mouth ulcers. But if you don't go to the doctor at that point, then it goes into that dormant stage and it can become chronic. So there's often no symptoms at that stage because it's just lurking behind in our CD4 cells. It's not doing anything. It's just hiding out there. But eventually it will become active and it will start destroying T cells, those CD4 T cells. And then it progresses to that syndrome that is called AIDS, which is acute immunodeficiency. Oh, I said immunodeficiency system. That should be acute immunodeficiency syndrome. And so this is considered the late stage of HIV infection. Your immune system is badly damaged and it's not going to come back. So you get more and more severe infections. So there is an important distinction here. HIV is not AIDS and AIDS is not HIV. So HIV is the virus that infects us, and AIDS is again, I put immunodeficiency system here. Sorry, it's acute immunodeficiency syndrome. So that's my error. I apologize. But that's the constellation of symptoms that is late stage infection from HIV. Here's just a graphic that shows you HIV is the virus, CD4 cell. That's what that's the captain of our immune system, so to speak, coordinating all the other cells to do what they're supposed to do. And then AIDS is the loss of CD4 cells and the increase of HIV virus in the body that just continues to assault us. So how is AIDS transmitted? Well, it's transmitted via body fluids. You probably recognize that at the beginning when I was talking about who gets it. Semen, preseminal fluid, rectal fluids, vaginal fluids, breast milk. Anybody who has HIV and is transmitting or comes in contact or excretes or those fluids come in contact with someone else, they can infect them. So some areas that you can obtain HIV infection are inside the mouth, through the penis, through the vagina, through the rectum, through any damaged tissue that's been cut or scraped, even if it's, like let's just say on your armpit or something, if you come in contact with those other fluids. And then also injection into the bloodstream. So if you are a drug user, or if you share needles, or if you're a hemophiliac, those are ways you could also get it. The blood supply is now very carefully screened for all transmissible diseases, but just so you know. So the actual routes of infection, or the common routes of infection, are vaginal or anal sex with someone who has HIV, who doesn't use a condom and is not on HIV medications. Talk about those in a minute. Or through needle sharing with someone with HIV. And some uncommon ways are from mother to child, through blood circulation, a nursing mother who can transmit it to her child through breast milk, and then needle sticks or other sharp objects contaminated with HIV. And this isn't needle sharing. This is like when you're working in a healthcare setting, and you accidentally stick yourself, or a scalpel cuts you and the person has HIV. So mainly healthcare workers. HIV is not spread by sweat, saliva, urine, any casual contact, mosquitoes, pets, participating in sports, touching things that were touched by, I mean, unless you have an open wound and you touch something that, has blood or body fluids from someone who has HIV, or eating food handled by a person with HIV. It takes a body fluid, a real body fluid to actually spread it. So this is a chart of, the prevalence of HIV. If you look at the left side of the chart, the darker colors are the number of people living with diagnosed HIV. The lighter blue is the number of people with undiagnosed HIV infection. So you can see in 1985, there were very few people who were diagnosed with it and a lot of people undiagnosed. But if you scoot on over to the right-hand side where we get to, about 2010 maybe, there are, perhaps 80% are diagnosed and another 18% are undiagnosed. So, and then the line graph just shows, that in another way that, the number of undiagnosed has gone down considerably. So here is another chart of people living with HIV. If you look over at the far left, 1980, that's when the virus first started coming around. The kind of oblong blue at the bottom are the number of people who died from HIV. And so you can see the red dots. There are a number of people living with HIV now, and there are fewer people dying than in the early 80s, where a majority of people died. So what's the treatment? In 1996, there was a huge advance, and there was a drug combination called HAART, and it was a combination of several drugs which produced a highly active antiretroviral therapy. Remember, this is a retrovirus, so you want to have an antiretroviral therapy. So these drugs just worked together to stop the virus from killing as many CD4 cells. It wasn't a cure, but it was a combination of many drugs, and it did decrease how quickly HIV killed people. And in 1997, so 15 years after the beginning of AIDS and a year after the development of this particular therapy, AIDS-related deaths declined by 47%. So there's also treatment. In 2012, there was pre-exposure prophylactic medicine. So if, for instance, you live with a man who is HIV positive, then you can actually take a pre-exposure prophylactic medicine. So if you take this correctly, it prevents you from getting HIV from your partner. Then, if you are exposed to HIV, you're not taking a pre-exposure medication, you can take a post-exposure prophylaxis. And this is often what healthcare workers do. So if they get a needle stick or something, they begin a combination of antiretroviral medications hopefully within 24 hours, but it can go up to 2 or 3 days. And they take this medication for 28 to 30 days, and that typically prohibits or prevents HIV from infecting any CD4 cells. So you never actually retain the virus. So what's the prognosis of those living with HIV? It's very good. So if you look at this, the very first one, a person without HIV, the average life expectancy is 79 years. A person with HIV diagnosed at age 20 who takes HIV medications can live up to about 71 years. And it may even be longer as we go forward with, the HIV medications. If a person does not take any medications or remains undiagnosed, then their lifespan is about 32 years. So they're not going to live very long. That's about 12 years after diagnosis.